Okay. Can you hear me? Yes. Okay. Welcome. It's a pleasure for me to welcome you to this special symposium organized by the Department of Physics and by the Quantitative Biology Institute. You're going to hear 6 talks of given by fantastic junior faculty about the cutting edge of what's happening at the interface between biology and physics with a focus on theory. And we have a very packed schedule. So I'm going to dive right in. And so the first speaker is Marianne Bauer. Marianne got her PhD in 2015 from the University of Cambridge in the UK. At the time, she was working on ultra-cold gases in low dimensions. And then she moved to Munich, where she worked with Erwin Frey to study the role of spatial heterogeneities on the extinction behavior in metapopulations. And then from there, she moved to Princeton. From 2018 to February 22, she was a HHMI postdoc and a fellow at the Center for Physics of Biological Function. And there she worked with Bill Bialek, Wischhaus, and Gregor, what was his first name? Thomas Gregor. And she was studying basically information processing in the regulated network of Drosophila. And then since March 22, she has been an assistant professor at the Department of Nanoscience in the University of Delft in the Netherlands. And so welcome, and we look forward to your talk about sense and decide information processing in gene regulation.
Yeah, thanks very much for having me here. It was really nice to meet everyone yesterday, and I'm also looking forward to the rest of the talks in the symposium. Yeah, I'm a little bit worried that the Zoom thing is going to be problematic. Let me just close this. Okay, so what I'm broadly interested in is questions on the accuracy and questions about the accuracy and efficiency in biological decision making. And actually, is the Zoom still working? I'm gonna just keep. Okay, good. Okay, so yes, it is on. I can move it higher. Is it better now? No. No. It's supposedly on. Okay, no, that's fair. Now, maybe a bit. Okay, so I'll try and hold my head. Yeah, so I'm interested in accuracy and efficiency in biological decision making. I come from, or I started being interested in this because I've done a postdoc in microbial model systems where I looked at ecological model systems and population dynamics. And there I started learning about the importance of spatial structure and clustering in these systems, and also sort of how the spatial structure is being maintained. I then did a second postdoc, as Thierry already said, on fly transcription, where I started learning that, information transfer ideas can help you learn about how the enhancer architecture works in these systems. And so because of that, and I'll hopefully show you in this talk that because of that, I've become interested in sort of what tricks biological systems can use to improve accuracy, because it's often not clear whether a particular feature that's there in the system reduces the accuracy or increases it. And with that, since I'm talking about gene regulation, I wanted to make sure everyone is sort of on the same page. So gene regulation describes the process with which genes are expressed that occurs because transcription factors bind to certain regions of the DNA. These regions are called enhancer regions, especially in eukaryotic systems. These enhancer regions have binding sites, and then the transcription factor binding recruits the polymerase and leads to the gene being transcribed. Often these cells need to respond to quite sensitive changes in transcription factor concentrations. And so we started being interested in, what aspects of this complexity really help with the precise sensing of the transcription factor concentrations. Concretely, this complexity that I have in mind is the number of enhancers. It's often true that several enhancers regulate the same promoter. Often they are several kilobase pairs away from that promoter in the genome. So there are these complex topological structures. The concentrations are often not homogeneously expressed. And so because of that, you know, I really wanted to highlight that these enhancers are important. The fly doesn't develop without it. They also make up a reasonably big part of the genome. About 10% of the non-coding genome of the fly is enhancers. And so understanding them a little bit better, I think, is really important. In principle, when you want to do that, there are two ways you can approach such questions. The first one is you have a model in mind, and the second one is you have an optimization goal in mind, and you try and learn how an optimal system would solve that problem that you have in mind. On the level of models, when it comes to gene regulation and early fly development, there are sort of these two very characteristic models. The first was developed in the 1970s, and it's called sort of the French flag pattern. So the idea is here that you have a transcription factor gradient. A gene turns on when the transcription factor concentration is expressed higher than a certain level and off below. So you have a strict threshold which determines the measuring of that enhancer. And the second class of models that I think is broadly developed much more now, also in the bioinformatics center, what I would broadly phrase as occupation models. So here you sort of consider what is the binding probability of a particular transcription factor somewhere on that enhancer. And then as a function of external concentration G, the amount of occupation of this enhancer goes up. So this variable C goes up as G goes up because individual molecules bind. That's now being developed by a lot of people around Rob Phillips and very mechanistic, but yes.
Can you share on Zoom?
Okay, I'll try this again.
Wi-Fi, yeah, she's falling on Wi-Fi.
Yeah, I don't know why. I am there. Oh, I see. I just need to reshare.
Thank you.
Okay, yes, of course. Okay, so okay. So I guess so far I was sort of recapping these two models, and I was saying that for the complexity that's present in these systems, maybe starting with models is a difficult idea because you would have so many parameters, especially if you have so many enhancers and so many binding sites. So there's this other approach where that's essentially model free. So there you assume that these enhancers fulfill a certain function, that there are certain constraints, and you use the data that you have available or that my experimental colleagues would have available in order to infer an optimal device that you then compare. So what I would like to do in this talk is that in the first part of the talk, I would like to tell you that this approach works. I'll just remove this microphone a little bit. I will hopefully convince you that this approach is going to work. You know, that basically working means that information optimization is a valid goal that, you know, can help you learn about the early fly embryo. I will particularly show that because of this information optimization, we can derive that there is a set of enhancers with a series of binding sites necessary for this process. And then in the, while always connecting to the models. And then in the second part of the talk, I will discuss a little bit how much fine tuning is required and linked to microscopic models of enhancers. So let me start with the data. Gene regulation in the early fly embryo is important because the fly is essentially left alone to develop individually. So basically the mother deposits some signaling molecules, in this case in the front of the fly. These signaling molecules diffuse, and sort of 2 hours after the egg was laid, they give rise to the expression profiles of the gap genes, which you see here in the middle panel, and of the segmentation genes, which you see here in the bottom panels. The important part of the segmentation expression is that they allow each nucleus along this embryonal axis to take a distinct cell fate. And so what we will do is that we will focus on, so they will take a distinct cell fate, and the enhancers that reach the gap transcription factor profiles are sort of order 50, and they have this complex binding site arrangements. And so we will try and focus on the gap transcription factor concentrations as an input, and we will try to find out why these enhancers are so complex. The gap transcription factors concentrations were measured by my colleague Marianne and Thomas Gregor's lab in this collaboration with Eric Wieschaus. I should say also that the theory that I'm presenting was done closely with Bill Bialik. So these gap transcription factor concentrations give you information about the number of distinct cell fates that nuclei along the embryonal axis can take. And now we essentially say that the enhancers need to sense this information precisely, because if the enhancers make sort of mistakes in their measurements, they can of course never really pass the information that's provided in the gap genes onto the nuclei. So that's what we're asking. Yes.
So each of those profiles is a different gap protein.
Yes, correct. 4 of them, correct. Yes. Thanks. Yeah, I should say also interrupt, of course, when there are things unclear. And so we hope that the enhancer is sort of trying to optimize a particular question. So we basically say that the sensor or this enhancer should, know as much as possible from these profiles about the possible cell fates. But we also assume that the noise and the noise, because of the binding and unbinding of the individual molecules at the enhancer is unclear, so that we don't really have a sense for how noisy this measurement is. This can be phrased very conveniently in information theoretical language. So we can basically say, we want this enhancer wants to optimize the information about the nuclear cell fates, while we don't know what the capacity, so what the information at the enhancer with which it measures these input profiles is. If you don't know about the mutual information, that's not super important. The only thing that's important is that you basically know that it describes a probability distribution between two variables. If you have two variables and you measure one variable extremely precisely, high mutual information will tell you that you know the other variables very precisely too, whereas low mutual information will tell you that you don't know anything about the other variables. So it kind of is a little bit like a correlation. And so, you think now we want to kind of optimize for this information theoretical goal, and we want to optimize over all possible probability distributions that describe an enhancer. Models, of course, can also be phrased in probability distributions, but so we really want to ask, let's just, not assume a model, let's just assume what this optimal probability distribution looks like. And you might think that this is super hard, but it so happens that this goal is extremely well known also from machine learning. And so there is actually a way to do this for this particular goal. Concretely, the reason why this is known as machine learning is that this question of there being data available and there being a variable that describes the relevant part of the data, that's a really important thing that happens also, when you have sort of an image and the thing that you care about is not the image, but the image content. So in that case, you want to essentially compress the information in this image that doesn't give you information about the image content out. So in that case, you would try and compress essentially the background definitely away. And then by looking at that compression, you can hopefully learn what the system does. This is what we will do here for the gap concentrations as an input and the nuclear cell fates as an output. And so with that, I would like to start with the data. And so I want to do this for one of these gap transcription factor profiles first. So you see this one here, it's hunchback, it's expressed along the embryonal axis. And what we will now do is that we do this compression, we sort of optimize for this optimized sensor, and we plot the traces of this optimal sensor in the information plane, which you see here in the middle. On the x-axis, you have the information between the sensor and the gap gene concentration. So that's basically the capacity of the sensor. If that is low, you have a lot of noise in the binding and unbinding. If it's high, then the sensor measures very precisely. That's on the x-axis. On the y-axis, you have the information that the sensor can give you about the possible nuclear cell fates. So that's the thing that you want to have as high as possible. And you can sort of see that there's a shaded area in this plot, which is because there's a maximum which tells you that you can never sense more precisely than there's information available. Similarly, the data processing inequality also gives you the other curve. But so the black curve that we see here is sort of the thing that we get when we do this compression, when we look at the optimal sensor. So you see that that is very close to the very optimal boundary. So that suggests that sensing in this case is possible, which is actually not the case when you do this analysis on different types of measurements of neuronal data, because there you often subsample so that the inference of a precise sensor is more difficult. In order to understand it, we can now go back to the sort of bottom-up models and compare it to the thresholded sensor. So if you remember, this thresholded sensor basically, in this case, would basically mean let's put the threshold on, and the enhancer can tell if the expression profile is expressed higher than that threshold or lower. So in this case, that would distinguish two sets of nuclear cell fates, the one on the front half of the embryo and the one on the back. Now you can do this with more and more thresholds. So you can now, identify more and more different regions of this embryo along the embryonal axis, and you see that after about 7 thresholds or so, this thing starts saturating. So that means that you need about 7 thresholds, and you can sort of then throw the thing that you just learned from the model on the information plane and compare it to this optimal sensor, and you find that it's exactly on top of it. So that I think is actually quite important, because it shows that this intuition that biologists had had for decades, that thresholded sensors would be good if they were implementable, that is actually consistent with an information theoretic goal. And therefore it makes sense to analyze this information theoretic goal a little bit more mathematically, because it's sort of more precise. It's like a more precise phrasing of that intuition that was sort of around anyway. And so that's what we want to do from now on. So I've already said that, we've looked at this and we found that 7 thresholds are necessary. So that already suggests that more than a single enhancer is needed. But let's look at it for all of the gap transcription factor profiles together, because of course these enhancers need to sense all of them together. So if we do the compression again, you see that, the plot is now slightly different. There's more information available. And you also see that in order to get sort of within 10% of the maximum of information available, you essentially need a sensor that has about 50-ish thresholds. And 50-ish thresholds, I think it's really hard to argue that that can be fit into a single enhancer. Even if you assume, and we'll come to that in a little bit, if you assume that this enhancer has a series of sort of strong cooperatively binding sites, then maybe, maybe if an enhancer has 10 or so binding sites, maybe it can fit like 2 or 3 thresholded measurements, but it will never be able to fit 50. And so the fact that we need 50 to sense all of these gap transcription factors means that the fly actually needs to have all of these different enhancers. Similarly, we can now sort of push this a little bit further and say, well, what if we break these enhancers up? So we knew we needed more. Let's assume we take 4, and let's assume we take the 4 in like the slightly awkward way where essentially one of the 4 can sense the transcription factor input for one of these gap genes. So we have one for a giant, one for hunchback, and so on. We optimize that, and what you now get is this blue line in the information curve, and you can see that that blue line for essentially all values of sensor capacity. So no matter whether your enhancer is precise or imprecise, it's always lower than this optimal line for all values. And that means that this type of splitting is actually really a terrible idea. And of course, the fly doesn't do that because the fly has enhancers that have binding sites for all of these transcription factors concentrations together. And I think that's also really important because when I started out on this project, my biological colleagues always, sort of asked, like, what do you want with your optimization approach? Like, biology is so messy and so redundant that, like, it's not optimal. Like, what can you actually learn from this? But I think what it shows us is that there are aspects of the complexity that are in a way consistent with optimization, that there are sort of, a lot of strategies that would not be optimal. And so we can learn about, how things should be structured in order for them to be really good at doing particular tasks. So I thought that was really exciting and really encouraging. And so that's essentially the first part of the talk. And now I would like to kind of discuss a little bit the questions that I've been working on since, which are sort of related to a lot of fine-tuning of these enhancers and also a little bit more of the microscopic models. So let's talk about fine-tuning. And in order to do that, I want to go back to the thresholds. But let me first say that fine-tuning in that phrase, we essentially want to relate to this discussion that has often occurred in biology about robustness of systems. It's just that fine-tuning is a little bit of a better phrase because it describes more precisely that robustness is often associated with having a parameter at a particular value or having a range of parameters available that you can tune through for the system to still do the same thing. So what we're doing here is that we now look at the optimal position of the thresholds, and we want to sort of see what happens if these thresholds move away from the optimum. How fast do you sort of fade out? And the way you do this is that you basically look at the curvature, so at the Hessian, but practically you look at the curvature in different directions around the optimum. So you are at the optimum, you tune a threshold, and you sort of see how fast does the landscape, or like how fast does the landscapes fall off? So how much information do you lose as you tune that particular threshold? In order to do that, you calculate the curvature, you then look at the eigenvalue spectrum. And then what you can see is that these eigenvalues are actually sort of, they span several decades. Sort of, therefore, especially for the system with many thresholds, there are some that are really, really flexible. These flexible thresholds happen to be the one at high concentrations, and you can sort of intuit that already by looking at that, at that transcription factor signal. So you can see that when that is expressed at high levels, there's actually a lot of noise in the system, and so it doesn't really matter whether you put that threshold at a particular value or at a particular slightly shifted value, because it's sort of going to pick up on the noise anyway. And so that's why these thresholds, the higher ones, you don't really need to put them into the right position. And I think that intuition is also around in biology, where there's often this intuition that the weak binding sites, which are the ones that are meant to measure high concentrations, because because I guess that's what they do, their arrangements, that doesn't matter so much. So their number and their arrangements don't necessarily matter. So again, this sort of explains something that's around in biology as well.
shaded areas correspond to the error bars?
Yes, that's right. It corresponds to the error bars, and the error bars is sort of calculated with respect to the data that we had available. So we had about 34 embryos. We look at a particular time frame where this transcription factor is expressed, and then we do subsampling essentially. So we, we look at what we get for, say, 10 embryos, 15, 20, all the way up. And then the mean is calculated by extrapolating, and then the standard deviation of this sort of population of subsampling gives you the error bars.
For example, the Gaussian will work.
Yes. Yes.
Can I ask what you mean by arrangement?
By arrangement, I guess by arrangement, I would in principle mean the placement, like whether, there are two next to each other or there's a sort of a wider gap. Yeah. Okay, and also sort of more theoretically, we're interested in this because it sort of more abstractly also connects to work that we're currently doing also with Kamesh, who's in the audience, where we're sort of saying that information theoretical optimization, so this is also with Bill Bialik, of course, and other people, but where we're saying that entropy or information theoretical optimizations often help you find landscapes where these sloppy directions are present. We know, and Ben in the audience is also a specialist on these sort of sloppy models, but so we know that sloppy models often occur in biology, that directions are often sloppy, so that means that there are often parameters that you can vary a lot. But when you sort of pick the wrong model, it might look in that wrong model that you've picked, it might look as though you can vary a parameter, or all of them are sort of equally variable. That's sort of a lot of work that's going on in statistics at the moment, because you know this from neurons. So you know that often models where you basically say that two neurons are coupled are similar to basically applying spins on each of the neurons, because basically you can increase your number of parameters and get the same sort of response. But when some of these models will not have this sloppy architecture. And I'm super interested in this. And I also think it's really nice because I think, with these information theoretical optimizations, you often find degenerate solutions at the optimum. And I think seeing that degeneracy is part of the intrinsic structure of the problem and not at odds with the idea that you've just done an optimization, I think is really crucial, and maybe also helps with the connection to biology, where, this entire idea that, there's one optimum, I think is sort of, understandably incredibly unpopular. And so I really like the sense that, we can sort of find that there is a degeneracy at the optimum. So now I would like to sort of also think a little bit more about what there was a questions also about these weak binding sites, and sort of think a little bit more precisely about, can we use these methods to actually predict something about enhancers? Predicting at the moment is really exciting and important, because there are several experiments where colleagues can vary the exact enhancer architecture. So for example, what you see here on the left is a collaborator of mine in Bonn, who is able to tune through randomly. So you see this big red block. What you see here is essentially the DNA sequence, different colors denote different base pairs, and this red segment is essentially thank you. This red thing here is essentially a block of G base pairs that he just shifts through the enhancers, and where he measures the output of that enhancer. And so we're trying to work on predicting these structures. Similarly, Angela De Pace's lab at Harvard has an enhancer for this particular gap transcription factor, actually, that has 6 binding sites. And so she can cross out these ones are sort of, the light ones are the weak ones, and she can sort of delete them and then again measure the profiles. And what you see here in these little, or what you meant to see here in these little figures, is that basically the blue one, which is the one where she has only one deletion of a weak binding site, and the blue one in here, where she has 2 deletions of a weak binding sites, are incredibly similar. And not only are they incredibly similar, but also when you really sort of look at the amount of binding sites that there are versus the Hill coefficient. I'll explain that a little bit on the next slide. So that essentially gives you how active is the enhancer, or how easy is it to activate the enhancer given another transcription factor. And if that thing is very steep, that's typically associated with many cooperative binding sites. If that's low, it's typically associated with only a few cooperative binding sites. But what's interesting, it does in principle in all of these heuristic models, you would assume that with the number of binding sites, that Hill coefficient increases by 1. What you see in this graph is that it does not do that. It actually very significantly increases, like a factor of 1/2. And Angela De Pace and her collaborators have, of course, done done, very precise stochastic modeling of these enhancers. But what we were wondering is there a sort of a simpler system that can capture the sort of lower cooperativity in these enhancers that she can measure? And so I just want to quickly explain this again. So this is in relation to this old model work, or not necessarily old, but actually very recent model work on these occupation sensors, where you basically get the occupation increasing as a factor of the incoming transcription factor concentration. You can do a lot of modeling, but you will always find that the steepest slope that you can get here is when you have cooperative binding sites. So when you have some sort of Hill function activation, and it's that Hill coefficient that they measure in these experiments. Now, the problem is that when you have these cooperative binding sites, early work from the sort of 2000s was able to estimate noise around these binding sites. And, then people have worked a lot on the precise formulation of this noise. They said that it depends on the binding rates, the diffusion rates, the measurement time, and other things, but essentially this noise always gets worse the more and more cooperative you get. And so people, especially a lot of the people that I talked to, sort of said, why would you use this cooperativity if it, makes the noise consistently worse? And so what we are currently trying to do is that we are sort of using these stochastic lattice simulations that I've gotten used to from my previous postdoc in order to simulate, what is the occupation if I basically have a small lattice here and sort of 3 binding sites for transcription factors, and then have transcription factors that can appear and disappear. First of all, we asked, can we encode the cooperativity also in the clustering of the transcription factors? Clustering at the moment is a big debate in the field because of this focus on liquid-liquid phase separation, where people say transcription factors can cluster, polymerases can cluster. And so the initial idea was that you can basically have cooperation between the binding site just because these transcription factor clusters. So that was one of the things that we tried to check, and then we also tried to check how much does the noise goes up. And what we actually found in these sort of preliminary work that a student of mine is currently working on, and also Ned Wingreen is involved in. So what we actually found is that quite surprisingly, what's more important, much more important than any of the clustering, because we are at such low concentrations for our transcription factor, it's actually much more important that the measurement time influences your noise much more than the clustering. Like even if you ramp up the clustering a lot, it almost makes no difference. So that's the 2 curves that lie essentially on top of each other. But if you increase the measuring time, you can really bring the noise down. And I think that's sort of important because it highlights this part that this question that it's sort of unclear whether clustering in this case contributes to bringing the noise up, or whether it's actually something that's helps with the cooperativity, or whether it's something that makes almost no difference at all. And second thing that I just wanted to highlight, going towards the end of my talk, is that we then try to zoom out again. So this is again now work that I've done with Bill, but where we started to think, well, what do we do if we sort of don't think about this cooperative binding, but if we just assume that the enhancer probability distribution is some sort of Gaussian distribution around the mean, and we perform our bottleneck optimization, we could then find that we can sort of simplify this bottleneck optimization drastically, and that in fact we can tune along this entire bottleneck curve that I explained before with the number of thresholds on it and so on. We can tune along this curve just by essentially changing the noise at the enhancer. So, these different thresholds would essentially correspond to having a different mean in this enhancer activation profile. So, you would have a threshold, a type of mean. But here we can show that we can essentially start with one C bar that we pick optimally, and then we tune along this curve. And what this means, I think, is also potentially exciting, because I hope that sort of all of these things will hopefully push the analysis so close to the experiment that we can eventually decide how close to optimality these systems are. So here concretely, because of this prediction that, in the low noise regime, so in the high information regime, because of what we know of this relationship, we would predict flatter profiles than we would predict in the sort of high noise regime. And so basically the hope is that you can measure an enhancer, you can measure the noise, and you can see, does the noise now correspond to, one basically, does the noise correspond to tuning essentially along this optimal information curve, or are we in a regime where the noise is essentially so high that that's then consistent with the steep activation profile? So I'm really hoping that this, that sort of measuring the profiles and trying to find out in which regimes for the information optimization a particular thing is optimal can give you sort of quantitative information about the biology. Then what sort of a small and nice sort of side effect of that work is that we were also able to, express the information goal entirely parametric. So that means that for all possible Gaussian input profiles, where we initially have all sorts of possible profiles in the information plane, we can collapse this because these things now only depend on the information that's provided in the profile and a single parameter lambda. So we can collapse this on the information plane, and we find this scaling collapse, which of course is very convenient, not only from the theoretical side, because you now don't need to do this extremely complicated bottleneck optimization. You can just sort of predict what a particular sensor at a particular capacity will do. But then also, again, practically, because it sort of shows that, if you assume that an optimal sensor is maybe located here, where it loses, where it gets the most, information out, but it's still not in the saturating regime, so it's not measuring overly precisely, then it would lose half a bit. And so losing half a bit of an information profile that has 4 bits of information is maybe not terrible, but if you lose half a bit of an information profile that has 1 bit of information, that's a bad idea. And so the argument is that these Gaussian sensors, like if you assume that, enhancers really measure sort of Gaussian probability distributions, then it makes sense for you to have an input profile that's not a steep threshold, because you will lose the sort of information that's the half a bit information because the Gaussian sort of has this intrinsic smoothness. And I just wanted to quickly sort of end on saying that we also try to push this work now towards sort of inferring 2 sensors. So you will remember that initially we said multiple enhancers are required. We tried to split it into these 4 enhancers, but we had a particular assumption for what these enhancers should look like. Now, what we try to do is that we say we don't have this assumption, and we just try and find what sort of 2 sensor combinations are optimal. And what's interesting here is if you basically imagine that you have one particular enhancer, in this case, with a certain capacity that can measure in 4 types of thresholds. You can split that, of course, into an enhancer that has, into 3 enhancers that have each 1 threshold, or you split it into 2 enhancers where one of these enhancers has a non-monotonic activation curve. So that's the same, but this middle part is in total capacity actually much higher than this part. So if you say that you have a capacity optimization constraint, these non-redundant profiles actually make a lot of sense. And indeed, and I think here the comparison to experiment comes a little bit sketchy, because the experiment has this network of transcription factors. But in principle, I think we can see that there is this sort of non-monotonic sensor activation where genes can act, or transcription factors can act both as repressors and as activations in certain regimes. So we also think that this sort of multiplexing idea is maybe also, can maybe also help us understand how the enhancers are structured. And so with that, I think I want to sort of wrap up and talk a little bit more about future directions. So I hope I was able to convince you that these information theoretical goals were able to help you understand a little bit more about the enhancers in the fly embryo, that we needed many of them. I hope that I was also able to show that fine-tuning of these enhancers was not required, and that in fact, also it's really important to look precisely at microscopic models for these enhancers, because we could sort of try and understand how the role of clustering influences the coercitivity and the noise, and we can sort of hope to infer that certain activation profiles are more likely in particular noise regime than others. And so I also talked about these multiple sensors, and I just quickly want to talk about temporally varying profiles, because that's something that's become very important to me recently. That work is also definitely not finished yet, but I thought I would connect it to people in the audience. So the idea here is that these nuclei obviously don't just sense static transcription factor expression profiles. The profiles change, and also the response of the nucleus changes. So what I hope, or what I've been trying to learn about, is sort of old models of exploration-exploitation trade-offs that are currently also important in machine learning, and also in these statistics questions, and one of these models is this optimal bandit problem, which is kind of sort of funny. Like, you imagine like an agent that basically has different gambling options accessible to it, and the agent would like to now find the machine that gets the agent the highest reward, and that's a sampling problem. So the agent basically needs to sort of, you know, play a machine, sort of see what it gets back, and so on. And in this case, of course, you would assume that maybe that's a little bit similar to what the nucleus has to do. So the nucleus has to pick one particular cell fate. It can sort of, you know, initially maybe measure the transcription factor concentration a bit wrong, do a bit of a wrong response, then, you know, it might measure again, it might get it right, and so on. And so we are also, and so in comparison to this method, to this optimal bandit problem, the interesting thing is that for optimal bandit problems, it's very clear what an optimal solution looks like. Precisely, an optimal solution in these problems is known to scale as square root of t. So that means that you basically look at how fast, or you would hopefully be able to look at how fast this nucleus gets to the cell fate as a function of time, and then sort of see whether or not that optimality relates to this problem. Of course, this is much more complicated because, again, what we're missing is sort of the intrinsic part of the landscape. In these bandit problems, of course, there's no landscape between the different machines. So the agent can choose one machine and in the next case, the other. In our nuclei example, I think if the nuclei sort of has already done one measurement or one pick, it's much more likely that a guess in the vicinity of that particular pick is successful than it is a pick super far away, because that would otherwise imply that your first measurement was extremely wrong. So I hope to relate that. I mean, first of all, we've done, this is also a collaboration where what we're currently looking at is to sort of also guess the optimal strategy, or like do an expansion analytically that will allow us to infer the variances and the bias correctly. And I think I don't want to talk about this. So I wanted to just quickly say that in terms of future work, so what I'm really profoundly interested in are, you know, first of all, looking at better optimization goals and constraints, these temporally varying profiles, and then concretely the spatial structure around the enhancer. And I would like to do this in, you know, with a series of different systems where it's sort of already kind of becoming apparent that there's a huge difference between synthetic and natural systems. These include gene regulatory systems, where I have some collaborations also in synthetic contexts that really just put the individual genes into the cell. I'm also starting to work a little bit on organoids, which I think are super interesting because they have this this difference between sensing and external stimulus, and then also rearranging and sort of correcting, like the cells sort of move according to what stimulus they measured. And then finally, I'm slowly starting to get a little bit into neuronal systems, where the interesting question would also be whether we can sort of observe in a developing system whether a particular neuron is part of a particular group of neurons or different. So in this case, we have a collaborator that measures neuron in the culture and the cortex, and we hope that we can assign the neurons to one particular group. So I have collaborators to that, but of course, I would also be delighted to collaborate with people here on that. And so with that, I just want to wrap up and say, you know, that's the accuracy and efficiency in biological systems is like biological decision making is what I'm interested in, especially in the tricks. And I think that that's really exciting in the synthetic versus natural context, where, you know, it's sort of unclear, like we saw with the clustering or the cooperativity, where it's sort of unclear whether a particular component can reduce the noise in the system or increase it. And yeah, with that, I would like to thank my collaborators. The work that I've presented, the fly embryo work, was all done with Bill Bialek, and then on the experimental side, Eric Wieschaus, Thomas Gerger, and Mariella Petkova, who measured these expression profiles, and then other theory collaborations that I've touched on, and my funding sources. And with that, I would like to thank you for your attention.
Fantastic. Thank you very much. Questions here?
Thanks for the great talk. I have a question, two questions around the clustering part. So if I understand correctly, you mean clustering is a one mechanism to achieve the high field coefficient, because you need clustering to involve multiple bindings?
Yes.
Okay. Then when you say increase the measurement time will help, are you averaging over, like if there's some temporal fluctuation, you're averaging over longer than the fluctuation is?
So yes, so we assume that there are sort of no, so we basically have we simulate the concentration. So there are fluctuations because these transcription factors appear and disappear. And yes, we do have to simulate for longer we do have to simulate for a long enough time for enough transcription factors to appear. But the question is sort of, I guess I wouldn't compare it necessarily to the appearance time, or I mean, of course they have to appear multiple times, otherwise the measurement doesn't make sense. But it's mostly sort of like how much time in general does this system have available to measure. So I think, yeah. So essentially we average for a lot more than the fluctuations, but in both cases.
Thank you.
That was sort of a related question. So the data you're using, so the output for transcription, are you looking at the dynamics of some of these new reporters that could kind of, maybe you can help explain what's happening at the enhancer to the fact that transcription is not either on or off. It's even when it's on, it's pulsatile.
Right. So at the moment, we're not doing that. I think there's currently data from a collaborator that's becoming available that's looking at these bursts. And the hope would, of course, be to look at that, because I think one sort of disadvantage of this system that I've studied is that we sort of say that the output is just, being able to tell nuclear position. But I think what you're suggesting in this case, or like being able to tell the sail fate, but what you're suggesting would be much more interesting, which would be to sort of, really measure the mRNA and the burstiness, and then sort of say, can we predict directly to the mRNA and from that then to cell fate? So that's the long-term hope, definitely, yeah. And I'm starting to work a little bit on that with new collaborations, yeah. Yeah, sorry. Mm-hmm.
Hello. This may be a relatively basic question, but is there a reason why cooperativeness implies or causes the Hill function that posed a problem in one of your?
Okay. So I think so. Basically, the idea why it sort of increases is like sort of a very heuristic model. So in the Hill function you have this coefficient g to the h. And that h sort of just corresponds to how many molecules bind at the same time. So it's a super poor heuristic model where you assume that these transcription factors bind at the same time. So if 4 bind at the same time, you need to have more of them around. And so with respect to input concentration, that thing, becomes sharper. So you can, I think there's sort of simple examples of hemoglobin where you can really simple sketches. And then why the noise becomes more noisy is because then for each of these molecules, they need to arrive also at the same time. So it becomes a much more rarer event. I hope that answered. Yes.
So actually following up on that, on the concept of noise, I was trying to understand that in the context of the biology. Are you thinking about the noise as the probability that these different transcription factors bind due to their affinities to the site? Or are you thinking about it? You also talked about phase separation, and there's the concept of nuclear architecture. Like, I was just kind of curious to hear how you were thinking about it in the context of the cell biology.
Yes, so I think in the context of the cell biology, when I talked about noise mostly here, I meant with respect to arrival and binding and unbinding. But of course, liquid-liquid phase separation also affects the noise with respect to arrival. And there's this huge sort of problem in the field where I think liquid-liquid phase separation, some people have said, oh, this is great. It reduces the noise in the sense that it reduces the background fluctuation in a concentration pattern because you can sort of absorb redundant proteins into a liquid-liquid phase separated bubble. Here, the problem is exactly the inverse. Like if you have a liquid-liquid phase separated bubble and that concentration is fixed, you can no longer sense a varying concentration gradient. Yeah. Mm-hmm.
In the banded part that you talked about at the end, it suggests a very different perspective than the information theory one. And so related to that, there are 3 questions that you didn't really get to. One is, what's the reward? Number 2, how do you introduce cooperativity into it? And number 3, does it give you a handle on estimating how many molecules you would need? Yeah. Okay.
So I think the reward is maybe in vague terms the easiest. So I guess we would sort of say, there's a particular external concentration, and then we would sort of say there's now a right, output of molecules that this thing has to produce. So that the number of that right amount of molecules, that would be the reward essentially, and we would measure deviations of that from that right number as like sort of a penalty. Does the corporation come in? I think probably not on this level. I think I would see that as a more sort of abstract model that, is sort of seen on the basis of the entire nucleus rather than the individual binding sites. And then the last question I forgot.
Can you calculate the number of molecules?
I see. That's actually
In a randomized algorithm set.
That's a super interesting question. And honestly, I don't think I know the answer to that at this point. I think it would probably also depend on the landscape, but I think I don't know, but that's super interesting. Thanks.
So thank you again for a fantastic talk.